Hi everyone, today I'm going to be doing a presentation on the renowned Tony Pawson. So just to introduce him, signal transduction plays an important role in the communication of a cell between another cell. And when signal transduction is interrupted, this leads to diseases such as cancer and diabetes. And Tony Pawson was one of the early pioneers in signal transduction, specifically in the receptor tyrosine kinase pathway, and what proteins are involved and how they function. So just some background on Tony Pawson. He was born in 1952 in the United Kingdom and acquired a PhD in biology in London, England, 1976. Moved to North America and was working as a research fellow in California after he moved to Canada, first doing independent study in Vancouver and later moving to Toronto to work at the Samuel Bloomfield Research Institute. And here Tony Pawson would make his great discoveries on signal transduction and the receptor tyrosine kinase pathway. So his main discovery is most well known for his contributions on the receptor tyrosine kinase pathway, which involves ligand binding to a receptor, forming a dimer, and phosphorylating tyrosine on the dimer. And Dr. Pawson's main contribution had to do with the SH2 and SH3 domains, which interact with the receptor tyrosine kinase. And these domains consist of similar sequences, 150 to 300 nucleotides in length, and are found on the signaling proteins. And so it was found that the SH2 domain contains a peptide binding motif which binds to phosphotyrosine and functions in phospholipid metabolism, gene expression, tyrosine phosphorylation, as well as protein trafficking and other functions. And it has a bipartite site. One region is specific to phosphotyrosine, and the other finds other sequences. And you can see in the bottom right 2 different SH2 molecules and how they bind different sequences. And the SH3 domain is a protein binding motif found on the same polypeptide as SH2, allowing the formation of signaling complexes, and is involved in signaling and structural components, functions, and linking signals between the receptor tyrosine kinase and receptor proteins, as well as regulating these signals. And just some applications of his findings. There has been research in inhibiting the SH2 domain in order to stop cancer from growing and tumor growth. And this was actually unsuccessful because they couldn't isolate the SH2 domain and specifically inhibit it. But there has been work done on inhibiting the receptor tyrosine kinase in order to cause tumor regression. And so just to conclude, Dr. Pawson has made a large impact on the field of biochemistry through his work on signal transduction. His findings on SH2 and SH3 domain clarified how signals are relayed in the RTK pathway, and through his research, other scientists are able to develop new medications, and for that reason, he is one of the great Canadian biochemists.